Okay, let's talk a little bit about Mendel. So it's in the gene idea. So this starts our genetics chapter. So a little background on Gregor Mendel. It says Gregor Mendel's work with garden peas became the foundation of the science of genetics. Gregor Mendel was a Gregorian monk who wanted to be a teacher, and he had some plots of pea plants that he was growing at the monastery. Wanted to be a teacher, couldn't pass the teaching test. So he had been growing these peas in the meantime, and he had been trying to study the life of these peas, of these pea plants. And he was trying to figure out why they reproduced the way that they reproduced. What was making the ratios come out, or making the percentages of them come out like they were coming out? And so he went back to school, took some math classes, and one of the classes was a statistics class. And when he was doing statistics, he saw some ratios cropping up that were the same types of ratios he was getting with his plant crosses, as to like maybe how many had one particular color of flower versus the other, or seed shape. These were different ratios, and he saw that there were some ratios, and he found that he could use statistics to predict the outcome of his crosses. Well, he presented his work in 1866 to the scientific community, and they pretty much poo-pooed him because they're different disciplines. Math has nothing whatsoever to do with biology. And if he thought there was a relationship that he could show between math and biology, he was crazy. And so they kind of humiliated him. And they were studying Darwinism anyway at that time. So he was kind of hurt that he was put aside by the scientific community. And he pretty much died not knowing the contribution he had made. When he died, a lot of his work was burnt by the abbot at the monastery. So what survived basically was what he published. So he'd worked out some basic concepts that had no previous publishing, or nothing was documented. He came up with these ideas, and the wording has been changed a little bit, and the explanation has been changed a little bit, but his basic idea, his basic thoughts are in this. He came up with the independent assortment. He came up with the law of segregation, the concept of gamete development, and the concept of dominance and recessiveness. Now, he didn't use those exact terms. He possibly used dominant over something else, but he didn't know about independent assortment as being a word and stuff like that. But his writings described that idea. So the terms were basically given to it a little bit later. Here's a picture of him. Kind of a dapper looking little guy there. Here's one of the crosses that he did. He had pea plants, and in one pea plant plot, they were all purple flowers, and they always grew purple flowers. Generation, they always had purple flower plants. In another pea plant plot, he had peas that always grew with white flowers. Every generation they were always white. So he wondered, what would happen if I got the pollen off of a white flowered plant and dusted the carpel, which contains the eggs, of a purple flower plant? What would happen? And he did that. And you see he transferred the pollen from the stamens, which is where the pollen is, of a white flower to the carpel of the purple flower. The carpels matured into pods, so they had peas in the pods. He planted the peas, and they all came up purple. And he was wondering, how could that be? I put white pollen on the purple flower plant. How could they come up purple? Well, we'll see what he found out a little bit later. Okay, so let's look at this next slide. And here's what was happening genetically. The pea generation is the parent generation. Let me get my pointer back out. There you go. That's the parental generation. And here's his purple flower plant, here's his white flower plant. Now, that's going to be the appearance, or the colors. That's called the phenotype. And the genetic makeup is the genotype. Well, this is some stuff he kind of figured out, that these letters came later, but he figured out the concepts. The genetic makeup of the purple had two big P's. One big P came from the maternal, like the mom, and one came from the pollen of the other plant. So maternal and paternal contributions. The mom gave a big P, so did the dad for this purple flower plant. So gametes, remember the chromosome number is going to be reduced. So this is going to be reduced down the middle. And you see that all of the gametes are going to have big P because that's all that's there. So you could put big P and big P for the different types of gametes, but they're all big P, so they just put one here. The white flower plants had two little p's, and that's what made that plant white. A little p from mom, a little p from dad. It's a white flower plant. And the only types of gametes it can produce, remember they're haploid, so they can only have one of the p's, are all little p's. They're all going to be little p. So when he dusted the purple flower plant over here, the eggs had a big P, with the pollen from a white flower plant that all had little p for white. The resultant flower had both of the traits, big P and little p. Big P happens to be dominant over little p, so whenever big P is there, that's what you're going to see, purple. And little p is there, you're not going to see that. So all the plants were purple. That's what he got in his first cross. When they all came up purple, they were heterozygous. They had one of each of the alleles, one for purple, one for white. And he figured out the dominance and recessiveness characteristics of one trait being dominant over the other trait. That's what he called them. He didn't know about these being called alleles. He called them traits. So let's look at some definitions. A gene, the unit of DNA along a chromosome that codes for a single characteristic, such as, like we did earlier, a tongue roller or non-tongue roller. I have the gene for tongue rolling. I have the capital, I think they give it T. And my wife has the non-roller, the little t. It could be for, hanging earlobes, free earlobes versus attached earlobes. Okay, that's the gene. One parent had attached and one had free earlobes. One had long fingers, one had short fingers. One had freckles, one has no freckles. One has dimples, the other one has no dimples. One has curly hair, one has straight hair. So those are the genes. They code for a single characteristic. The Mendelian definition, they just talked about traits or colors. Now the definition is the unit of DNA that codes for a specific polypeptide. That's the molecular definition. That's the current one right now. Beforehand, it was characteristics. They were traded or blended between the parents. That's all they knew. Now we know that it does make a specific polypeptide. This is not said about which type of polypeptide because they're structural and functional. This one includes them all. For a specific polypeptide, it could be structural or functional. So this is the most current definition. There have been many definitions of a gene as time progresses, but this is the most current. Now, alleles, different forms of the same gene. These are what we use letters for. The letters are going to be the alleles. Like a big P allele was for purple flower, and the little p allele was for white. So the big P represented a gene that was going to give that flower purple flowers, or that pea plant purple flowers. And the little p was the gene, the allele, that's going to give that plant the characteristics of white flowers. The locus is a short term for location, the location of the gene on the chromosome. Now, locations of genes are a little bit different than measuring with a microscope because the chromosomes are so small. They have bands on them, B-A-N-D-S, little lines and stuff, and Usually they'll measure so many thousandths of a unit down, or they'll look at a particular band, and that's where they know the gene for something is in that area. The genotype, the actual genetic makeup of an individual. The genotype is an expression of the alleles. You know, the letters. We use the letters on the genotype, like big P, little p, big A, little a, big T, little t, that kind of thing. The alleles to represent the genotype. The phenotype is the physical expression or the actual expression of a characteristic, like, you know, freckles, no freckles, free earlobes, attached earlobes, tall, short, you know, that kind of stuff. Different color eye colors, hair color, that type of stuff. Tongue roller, non-tongue roller, whichever you want. But the phenotype is what the expression is. The expression of the genotype is the phenotype. Some other terms, dominant versus recessive. Let's look at dominant, an expression that's always expressed if its allele is present in the genotype. Well, in the case of the one we just saw, the big P is dominant over little p. If you ever have a big P, the pea plants are going to have purple flower plants. The only way you can have white is if it only has the recessive. So look at recessive. This is recessive alleles. An expression that is expressed only when its allele is the only form of the gene present in the genotype. The only way you're going to see white is if both alleles are little p. Little p, that's white. Big P, purple. Big P, purple. Little p, two recessives, white. Homozygous versus heterozygous. So you got a lot of definitions in this chapter. Homo means the same, hetero means different. So look at the definition. The condition in which both forms of the gene are the same. Big A from mom, big A from dad. Or little a from mom, got little a from dad. They're the same. Big A, little a, little a. Heterozygous, the condition in which the two expressions of a gene pair are different. Big A, little a, that's heterozygous. It's got one of each. Hetero, remember, hetero means different. Homo means same. So if you had big T, little t, big A, big B, little b, those are all heterozygous. One of each. We have different types of chromosomes also. The sex chromosomes, that's the 23rd pair of chromosomes, the X's and Y's. So the chromosomes involved in sex determination. In man, these are X and Y chromosomes. Remember, females have two X's. Got one X from mom in her egg and one X in dad's sperm. That made a female. If it's a male, the X was in the egg and the Y came from dad in the sperm. The individual that determines the gender is typically said to be the male because he can contribute X's and Y's to the gametes. Females, all of hers are X because that's all she has. Autosomes. Okay, autosomes, chromosomes not involved in sex determination, although they may carry genes for the secondary sexual characteristics. So the first 22 pair of chromosomes are called autosomes. The first 22 pair of homologous chromosomes are autosomes. They don't determine the gender, but they do carry genes that will bring out the traits of whatever gender that is. A karyotype, karyo means kernel, and in the olden days when they were first looking at cells, they saw a little kernel, a little nut in the center of a cell, and they called it, you know, the karyon. That's what we call the nucleus now. But they still, for, I guess, old time's sake, they still use the word karyo for karyotype. A display of the metaphase chromosomes, they've already condensed all the way, in which the homologs are paired together and photographed. So they'll get a metaphase cell, they will stop the cell division with something called colchicine, and then they will swish it or take pictures of all the chromosomes and then put them together. So here's a karyotype. Female karyotype. See, all the number 1s are parallel, number 2s are paired, 3s all the way down, and you get to the sex chromosomes, the last pair, she has 2 X's. Now the way they pair these, you can look kind of close. You look at the length and you look at the banding patterns. Look at the banding patterns of 11, 12, 10. You can see that, oh, look at number 7, that's pretty good. So you can see the banding patterns and the length. That's how they pair them up. And that's a female, 2 X's. Well, here's a male. He has one X and 1 Y. Now look at that Y. It's a lot shorter than the X. What that says is that the Y chromosome is missing some information that's found on the X chromosome. And that gets into sex-linked traits, where the X chromosome has the characteristic on it, but the Y is missing it. And so whatever mom gives the son, that's expressed. If mom has the genes here for normal vision, then the son has normal vision. If mom is passing the genes for color blindness, then the son is going to be colorblind. If she's passing pattern baldness, then the son is going to be pattern bald. That's what my mom did for me. That's why I'm missing some follicles on the top of my head. And another one is like a hemoglobin. I mean, not hemoglobin, hemophilia. Hemophilia is also sex-linked. Has to do with the production of, blood clotting mechanisms. Ooh, more definitions. This is one you're going to have to beef up on. Like a lot of definitions, guys. Characteristic. A heritable feature, one that can be passed to your offspring, such as flower color, eye color, height, ear shape. You probably know that you've passed characteristics off to your children, or if you look at your parents and look at your brothers and sisters, you can see that your parents passed off some of their features to you and your siblings. Those are characteristics. A trait is variation of a characteristic, such as a white or purple flower. So characteristic is flower color. I didn't tell you what it was, did it? But the trait is which color that is. Which eye color, height, tall or short, that's the trait. Ear shape, have your attached and you're free. So people will oftentimes use these synonymously because they're so close. This is a real, include all. It didn't tell you what color, it just got color. This one told you exactly which color it was. So these kind of get used together. True breeding in plants means when plants self-pollinate, the offspring are like the parents. Remember Mendel's first pea plants, he had plots of white flowers and plots of purple flowers. And every year the white flowers produced white, and every year the purple produced purple. Those were true breeding. Every year you get the same result because those are the only gametes, the only alleles you have in the population. The purple flower, all it has is big P's for flower color, and so they're always going to be purple. The whites, all they ever had was little P's, and so they're always going to have white flowers. Hybridization, making a hybrid, the mating of individuals with different expressions of a character. Now, when he put the white pollen on the purple flower plant, that's a hybridization. The offspring are going to be purple, but they have a heterozygous set of alleles there. So typically on hybridization, you'll see a blending of characteristics. That's what you typically see, which is why when you look at your brothers and sisters, you see that y'all have characteristics from both of your parents. So all of us are hybrids of our parents, and our children are hybrids of us and our spouse. An organism has 2 genes for each characteristic. Like, again, getting back to the tongue rollers for me. I have a gene for tongue rolling. My wife has one for non-rolling. Our kids have both of them now. They have my roller and her non-roller. The law of segregation states that members of a gene pair separate during gamete formation, meaning that each gamete receives only one gene from each pair during meiosis. That means that if the parent has big P, little p, they can produce two different types of gametes. Half will have big P and half will have little p. Half will have big P and half will have little p. Members of a gene pair, big P, little p, separate during gamete formation. Remember, gametes are haploid. Big P, little p, that's diploid. Each one of those is on a chromosome. Each gamete receives only one gene from each pair during meiosis. So the gametes are only going to have one of those alleles. And if you were big P, little p, half of the gametes are going to have big P and half will have little p. The concept of independent assortment states that the members of one gene pair of homologs segregate independent of all other homologs during gamete formation. Let's look at the next slide. That's what I was talking about with my pennies, and that's what we're talking about with this slide here. This is how they could line up, and it's going to be a pretty astronomical number that all dad's chromosomes that he gave you line up on one side and all mom's on the other side in meiosis to form the gametes. It's going to be a random pairing, random pairing like my pennies. They're not all going to be P's on one side and D's on the other. It's going to be a mixture, and that's what's called independent assortment. It's a random pairing of homologs, how they line up. More definitions. Monohybrid cross. Now, mono, means one. So, cross that involves only one pair of contrasting alleles, like flower color. Okay, because you could have the flower color is all you're dealing with. It's going to be either purple or white. But it's only one thing, it's flower color. A dihybrid cross has two different traits. It says crosses are cross that determines the inheritance of two different pairs of contrasting alleles. So you would have like flower color, big P, little p, and height, big T, little t. Big T for tall, little t for short. So you have flower color and height. That's two different pair of alleles. That's called a dihybrid cross. You have two traits there, two different alleles. And we'll do some problems in just a minute. A test cross. This again is done for plants, not for animals typically. Between an individual expressing a dominant phenotype, that's like, let's say, a purple flower plant. If you know big P is purple and little p is white, and you know that big P is dominant over little p, then a purple flower plant could be big P, which it is, or it could be big P, little p. It's still purple. So if you want a pure breeding purple flower plant, you want one that's big P, not one that's got big P, little p in there. So a cross between an individual that looks like it's got a dominant allele, dominant phenotype, big P, with an individual expressing the recessive expression of the same character. That means little p. So you know exactly what that one is, but you don't know what purple is. The cross is done to determine if the dominant individual is homozygous, big P, or heterozygous, big P, little p. And we'll come back and we'll see a picture of that in just a little bit. I'll show you a test cross, what that looks like, and that's what Mendel basically did. That's how he figured out what was going on with his plant. So a monohybrid cross. Okay, the first cross that you do is called the F1. So you have parents, which will be labeled a P, and then what we're going to get in here is going to be the cross, okay, of the plants. So when you do this, and it says in garden peas, purple flowers, big P, are dominant to white flowers. What I would do is I would, over here on the left, I would put big P equals purple, and then little p equals white. Okay, so you got your code on the paper in front of you. Cross a pure breeding plant with purple flowers. If it's pure breeding, it's going to be big P. With a white flowering plant, if it's white, it's got to be little p. That's the only way it can be white. Give the phenotypic and genotypic ratios of the F1 and F2. All right, the F1's the cross, the first cross, and the F2 is where you cross two F1s. So let's look at this. Here is here are two individuals. In fact, let's go back just a second. What's going on there? We're missing a. I guess I'll have to do this. Let's see if I can do this on here with this text. You'd put like big P equals purple, and then down below that you'd put little p equals white. Okay, so you have your code, and it says cross a pure breeding purple plant with one with white. So I'm just going to draw this. It's going to be a little easier to draw. Pure breeding purple, big P, with, that's a cross there, little p, Okay, that's white, because it told you that purple was big P and white was little p. Pure breeding purple, that's all it's got, big P. White, that's all it's got, little p. Now you know that the only types of gametes this individual can produce, I'll put a little, no, not that, I'll put a gamete right here. And I'll put one over here. Well, only type it can produce are going to have big P's in it. All the gametes can have big P. Now you could put big P and big P, but why? They're the same. And over here on the other one, the only types of gametes this individual can produce here, well, let me get down a little lower and get another one. Only type in here are going to be little p's. So little p. Make sure you put that little tail on there. And when they come together, when this sperm, I'll put a little, here it is, I'll make a little sperm there. When that fertilizes the egg, you end up getting a resultant fertilized cell called a zygote. Gosh, I gotta learn to get these things more accurately placed. It's going to have a big P and a little p in there. Okay, so one parent was purple, that's all they had was big P, big P. So all their gametes are going to have one P, and since they only have big P's, they're all going to have big P's. The white one, all it had was little p's, and so all the gametes are going to have little p. So when this one's gamete, and I made it a sperm, fertilizes this egg, the new offspring are going to be big P, little p, so they're all going to be purple. Well, an F2 is crossing two F1s. What's going to happen next? Here's an F2 cross. You're going to cross two of what you got on the F1. Now you see that there are two different alleles here, big P and little p. So half of the gametes are going to have big P, half are going to have little p. This individual, half of its gametes are going to have big P, and half are going to have little p. Now we use something called a Punnet square. It's this box here. And the way it works is, it doesn't matter which parent's gametes you put across the top or down the side. It doesn't matter. But one parent's gametes, like this parent here, let's put them down the side. Let's put them down here. So you put big P beside a box here and little p beside a box there. The other gamete, the other parent produced same gametes, but that's okay. You put them across the top. Big P on top of one box, little p on top of another box. So the alleles and the gametes go on top of a box from one parent and by the side of a box on the other. And then you just add them together. It's called adding the boxes. and that goes big P, big P. See, big P, big P. And for over here on the right hand side, big P, little p. And then down here, this big P up here, big P, little p, and then little p, little p. Okay, so it's called adding the boxes. And you get 4 boxes here that have different combinations of alleles in them. The box on the upper left-hand corner is the only one that has big P, big P. 1 out of 4. So down here, you have 1 big P, big P. If you look, you have 2 big P, little p's to 2 big P, little p's. And you have 1 little p, little p. So the genotype is the ratio of the different combinations of alleles. 1 of the 4 was big P, big P. 2 out of the 4 results were big P, little p, and 1 was little p, little p. That's the genotypic ratio. The phenotypic ratio is what you see. What you see. And what you can tell by looking at the genotype is that 3 of these are going to be purple because they have big P, and 1 of them is going to be white. So you get 3 purple to 1 white. The phenotype is what you see. 3 purple to 1 white. Now here is a dihybrid cross. A dihybrid has two traits. See, it says big P, yellow seed is big Y, green seed is little y. Okay. Then you see, oh, I don't want that one. I guess I could. Round seed is big R, wrinkled seed is little r. So you have 2 different traits. You have color and you have shape. That's a dihybrid cross. And again, over here on the side, I would recommend when you do these things that you put them out. I don't know how to get a bigger lettering here, but you would do big Y equals yellow seed. Oh gosh. Let me do that over again. That's in the wrong color. Let me get where you can see it. Big Y equals yellow. And then you're going to go little y equals green. Then you would do it again. Let's change colors. Let's do red. Big R equals round seed, and little r equals wrinkled seed. Okay, so that gets your code over here, and so you can look at it without having to read back through the notes. Cross a plant homozygous, that means the same, homo the same, for both yellow and round seed. So homozygous for yellow, round. Homozygous for yellow, that's going to be big Y, and round, big R, big R. Okay. With, here we go, crossing that with a plant that produces green wrinkled seed. Well, green and wrinkled, green is going to be little y, little r, little r. Is that correct? That's what it'll be. So let's look at the next slide. I would recommend putting this code over here anytime. Okay. Here it is. Here's the cross. Big Y, big R, with little y, little r, little r. Now, you remember gametes only have 1 of the 2 alleles for each trait. They're only going to have 1 Y and they're only going to have 1 R. How many different Ys do you have here? 1. They're all the same. How many different Rs? 1. They're all the same. So all the gametes are going to have one big Y and one big R. There's no other combinations of gametes you could get. Over here, you have 2 little y's and 2 little r's. The gametes are going to have one Y and one R. How many different YRs can you form? Well, you only got little y's, you only got little r's, so it can only produce one type of gamete also. They're all going to have little y, little r. When this sperm on the right fertilizes the egg on the left, let me get my hand back. Yeah, sperm on the right fertilizes the egg on the left. You just join the letters back together. The dominants usually go first. The capital letter goes first. So big Y, little y, big R, little r. You just pair them back up. So all the offspring are going to have big Y and big R. So what are they going to be? They're going to all be yellow with round seed. They're going to have yellow round seed. And the genotype, they're all big Y, big R, little r. So the genotype shows all the alleles that are there, different combinations of alleles. They're all this. You don't have any other combination. And the phenotype is what it looks like. Y for yellow, R for round. So they're all yellow round seed. Now this problem that they gave us on this exercise, I'm not going to give you one like this because the F2 is going to be what's called a heterozygous. That's heterozygous. Those are different. Dihybrid cross, 2 traits. Both traits are heterozygous, and there's a classic outcome for that. Here is, I just kind of put it all up here for you. The big Y, big R, little r. How many different types of gametes can it produce? Now let me tell you how you can figure that out just with numbers. How many different Y's are here? 2. So you would write 2 up there. How many different R's are here? Oh, there's 2. So again, 2. Now, 2 times 2 is what? Equals 4. So there's 4 different gametes that individual can produce. 2 different alleles here times 2 different alleles here, there's 4. And you can see that they're right here, these 4. Let me get a hand back. There we go. So here's how you can do this. First of all, find out the different combinations with big Y and the R's. So big Y in the gamete could pair with a big R. There's one, big Y, big R. What if the big Y paired with the little r? There's big Y, little r. So you've got the 2 pairings the big Y can do. Move over and see what the little y can do. Little y can pair with big R, little y, big R. Little y can pair with little r, little y, little r. And that's how you get the 4 potential gametes. But you knew you were going to have 4 because I showed you how to do it. 2 times 2. There are 2 different ones here. There's 2 different ones there. 2 times 2 is 4. Well, on this cross, this heterozygous dihybrid cross, I won't give you one like this because it gets 16 boxes. When you put one parent's gametes across the top, one above each box, and the other one down the side beside each box, and you cross them, you do big Y, big Y, big R, big R, big Y, big Y, big R, little r, and then you can trace over big Y, little y, big R, big R, big Y, little y, big R, little r, and you keep doing that, pairing them up all the way down. You end up getting a phenotypic ratio of 9 yellow round to 3 yellow wrinkled to 3 green round to 1 green wrinkled. That's the phenotypic ratio. That means that there are 9 that have a dominant allele for both, and I'll show you. I can't show you on this one because all the animations are taken away. But see, big Y, big R. That's 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 9 that are yellow round, dominant for both. 2, 3 that are dominant for the first and homozygous recessive for the second. So it's going to have little r's. dominant for the first and homozygous recessive for the second. That's 1. There's 2. And there's 3. And then there's 3 that are homozygous recessive for the first and at least 1 dominant for the second. And you start looking around, there's 1, there's 2, there's 3. And then 1 is recessive for both, wrinkled, green and wrinkled, and there it is right there. When you look at the different combinations, and again, this is why I'm not giving you this one, it's a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 genotypic ratio. Now, that's why I didn't write them out either, because it takes up too much space. There's 1 like this, homozygous dominant for both. There are 2 that are homozygous dominant for the first and heterozygous for the second. There's 1 that's homozygous dominant for the first and homozygous recessive for the second. There's 2 heterozygous for the first, homozygous dominant for the second. Here's 4 that are heterozygous for both, and it keeps going all the way across. That's just a lot of work. So not all dihybrid crosses are going to give you 16 boxes. I'll see if I can make something up that will show you some different examples, okay? Getting back to this test cross. Yeah. A cross between an individual expressing a dominant phenotype, the expression, what it looks like, with an individual expressing the recessive expression of the same character. The dominant would be in plants like the purple flowers. The recessive would be white. So you know what white is. You know it's got to be little p, but you don't know what purple is because it could be big P, or big P, little p. And so that's what it gave you here. In garden peas, purple flower plants are big P, are dominant to white flowers, which are little p. Is a purple flowering plant homozygous or heterozygous? You can't tell, not by looking at it. You can tell by doing a test cross, and that's what we're going to show you on the next page, is how you could tell whether something is homozygous or heterozygous. If the plant is homozygous, if it is actually big P, and you cross it with a white, they went ahead and put all the gametes. See, this can only produce 1 type of gamete, big P. This can produce only 1 type of gamete, little p. So you didn't have to really put 2 big P's and 2 little p's. You could have just put 1 and 1, because they're all going to be big P, little p. They're all going to be purple. So if it's homozygous dominant, all the offspring are going to be purple. There it is. But if it's heterozygous dominant, heterozygous, big P and little p, and the big P is dominant, then this one can produce 2 different types of gametes. Half will have big P, half will have little p. This one can still only produce 1, all of them have little p. So you didn't have to do the bottom set of boxes, really. This is 1 parent across the top, and the other ones, since they're the same, you just make 1 row. Half will be purple, half will be white. So that tells you where those whites come from. Well, this parent was carrying a white allele, and when it paired with the white allele from the known, you got white offspring. So it told them that this one was not homozygous. It was not pure breeding. And that's what a test cross is done for in plants. Now here is something, I couldn't have come up with this definition. The rule of multiplication. Listen to this one. The probability that independent events will occur simultaneously is the product of their individual probabilities. Wow. Here's an example. The probability of giving birth to a daughter is 1 out of 2 at any one birth. Now, females have 2 X's, males have a Y. So female can produce 1 type of gamete. Male can produce 2. And when you do a Punnet square, you put mom's X beside 1 box, and then there's 2 boxes across the top. One has an X and one has a Y. And when you solve that Punnet square, you get 1 XX to 1 XY, 1 female to 1 male. So that's 1 out of 2, or 50%, 1/2 at any one birth of a particular gender being produced. So you get a 50-50 chance of being male or female. Now that's probability-wise. The probability of having 3 consecutive daughters being born to a couple is 1/2 for the first one, times 1/2 to the second, times 1/2 to the third. That's 1 out of 8 families of 3 kids will have daughter, daughter, daughter. I have an uncle in Fort Worth that has 4 boys. So that's going to be 1 out of 16. 1/2 times 1/2 times 1/2 times 1/2. 1 out of 16 families will have boy, 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 boy. Now, if you think about this rule of multiplication can help you out on some other things too. If you have a die, how many sides does that die have? It has 6 sides. So what's the chance of you getting that die and rolling it on the table and getting a 3? That's 1 out of 6, isn't it? What's the chance of you rolling on there and getting a 5? That's 1 out of 6. What's the chance of you getting that die and rolling it twice and getting a 1 and getting a 1? 1 on the first roll and a 1 on the second roll. So you say, I'm going to roll it twice. What's the chance of me getting a 1 and a 1? That's 1 out of 6 times 1 out of 6. So what that says is 1 out of 36 double rolls of the dice, you should get a 1 and a 1 back to back. If your die had 8 sides on it, what's the chance of getting a 3? 1 out of 8. What's the chance of getting a 3 on the first roll and a 3 on the second? 1 out of 8 times 1 out of 8. 1 out of 64 times that you roll the dice twice will you get a 3 and a 3. And the more you do it, it comes out closer to the 1 out of 64. So that kind of gives you the way to predict stuff, how many different outcomes. Flipping a coin, what's the chance of getting a head? 50%, because it's only got 2 sides, a head and a tail, so it's 50-50. And so you can do the same thing. What's the chance of me getting a head the first flip and a head the second flip? 1 out of 2 times 1 out of 2, which is 1 out of every 4 double flips. Should you get a head and a head? Here's the rule of addition. Now this one's a little weird. The probability of an event that can occur in 2 or more ways is the sum of the separate probabilities of the different ways. Who knows what that means? Okay, this one is just saying if you were to have 2 parents and they're both heterozygous, big A and little a, and I'll see if I can make a slide and maybe insert this slide in your PowerPoint set or something, because I usually do this on the board. What's the probability of their having a child that is also big A, little a? So they want to know what's the probability of the A being from the egg and the little a in the sperm. Well, if they're both big A, little a, they produce 2 different gametes, right? Half will be big A, half will be little a. So the chance of getting them the big A from mom is 1/2. And in the dad and the little a, since he's got the same thing, it's 1/2. So 1/2 times 1/2 is 1/4, 1 out of 4. Well, then they want to know, well, what's the chance of the child getting the little a from mom, which is 1/2, and the big A from dad, which is 1/2. That's also 1/2 times 1/2 is 1/4. So you add that together, 1/4 plus 1/4, that's 2/8, or 2/4, I mean, which is 1/2. So the probability of them having a child that's also like them, big A, little a, is 50%, 1 out of 2, or 2 out of 4 if you do a Punnet square. If you put that big A, little a, and did a Punnet square, you would have a box that had 4 boxes, made of 4 boxes. One parent's gametes across the top, a big A on one top of one box and a little a on top of the other box. And down the side, a big A to the left of the first box and a little a to the left of the second box. And then you add them together, it would be big A in the top left box, and the top right box would be big A, little a. The bottom lower box, big A, little a, and then the bottom right box, little a, little a. So when you look at that, there are 2 of them that are big A, little a. 2 out of 4 is 1/2, 50%. Incomplete dominance, another type of dominance that you could talk about here. Incomplete, we've been talking about complete dominance so far, where big P is dominant over little p. And if the alleles ever have the dominant allele there, the big P is going to be purple. And the only way you can get white is little p, that's it. So one allele is dominant over the other. Well, this one's called incomplete dominance. Incomplete. Neither allele is dominant to the other. So neither one's going to be dominant over the other one. Thus, or therefore, each is expressed in the heterozygous genotype, producing an intermediate phenotype. And they give you an example. If a red-flowered snapdragon, which is what this is, it's going to have big R, big R. The alleles from mom and dad are both R, big R. And a white is R prime, R prime. They did R prime instead of little r's. But if you have a heterozygous form, big R, little r, it's an intermediate, it's pink. So when you talk about incomplete dominance, the heterozygous individual is a blending of the two extremes. In this case, a blending of red and white paint would give you pink paint, just like this. Now they want you to cross two pinks. So there's your R, R prime, that's pink, crossed with R, R prime, which that's pink. Now you can see that each pink individual can produce two different types of gametes. Half the gametes are going to have R, half the gametes are going to have R prime. And so you put that on your Punnett square, one parent across the top, one parent down the side, and the gametes down the side. And then you add the boxes back together. That's going to be big R, big R, big R, big R prime, big R prime, R prime, R prime. So that's what your Punnett square shows you. Now you find out how many different combinations of alleles there are. There's one, this big R, big R. There are two that are R, R prime. And there's one, this R prime, R prime. Okay, so that's where you get your genotype right there. Now the phenotype is what those look like. Well, you got the code up here at the top. Big R is red, so you got one red, to two R prime, two pink, to one R prime, which is white. So that's how you would do this incomplete dominance problem. It's worked the same way as any of the other problems we've looked at. In fact, all these problems are worked the exact same way. It's just that they're interpreted a little differently. This one is incomplete. The heterozygous form is a blending of the two extremes. That's what you call incomplete dominance. Codominance. Now read this and I'll give you my example. Inheritance characterized by the full expression of both alleles in the heterozygote. Okay, so you have shorthorn cattle. Red is RR, white is R prime, and the heterozygous R prime is called roan. Roan is a spotted cow. It has red fur and it has white fur. So both extremes are expressed in a codominant situation. When I was taking this, I just thought of something that would be easy to remember. And in an airplane, in the cockpit, you have two pilots, a pilot and a co-pilot. So they're both there. So when you have a codominant problem there, when it's a heterozygous form, they're both seen. So when you see a cow that is brown and white, that's a roan cow. If it's black and white, fur, that's a roan cow. So spotted cows are roans. Well, they gave you the code here. Red RR, roan RR prime, and white R prime. And they want you to cross two roans. So cross two RR primes. Well, you can see again, there are two different alleles there. So they can produce two different gametes. Each one can. The one on the left can produce half will be R and half will be R prime. The one on the right can produce half will be R, half will be R prime. So you put one parent's gametes across the top, R prime, one over each box, and the other parent's gametes down the side, R prime down the side. And you solve your boxes just like we did before. R prime. Well, there's R prime, R. You always put the R first. and then R prime, R prime. Okay. Get your genotypic ratio, the different combinations of alleles. 1 out of these 4 is RR. 2 out of the 4 are R prime. And 1 out of the 4 is R prime, R prime. That's your genotypic ratio right here. The phenotypic ratio is what you see. Well, you got 1 red to 2 roan to 1 white. Okay, so this is codominance. This type of a problem is called codominance. And the heterozygote, you see both extremes. Both extremes are there. Both are there, like the pilot and the co-pilot are both in the cockpit. So they're both expressed in the heterozygous form. Now we have multiple alleles. This is where you have more than 2 for a particular trait. In this type of inheritance, there are more than 2 alleles for the same characteristic in the general population, although any one individual has 2 genes for the characteristic, or you're going to have 2 of the alleles for the characteristic. And the ABO blood grouping is one of those, because we have blood type A, blood type B, blood type AB, and blood type O. We have 4 different combinations of blood type. So blood typing shows multiple allele inheritance. So it has 3 alleles. We're going to see their IA, IB, and little I for these 3 expressions here. Dominant and recessive, we'll see that A is dominant over O, B is dominant over O, and A and AB is codominant. They're both expressed. So multiple allele inheritance expresses multiple alleles, more than 2, dominance and recessiveness, and codominance. Let's look at the key over here. Here are the alleles: IA, IB, and little I. IA codes for type A blood. It's an antigen on the surface of the red blood cell. IB codes for type B, a B antigen on the surface of the red blood cell. And little I, type O, that O actually stands for 0. There is neither A nor B. So you don't have either A or B. That's what the O stands for. Well, let's look at the genotypes for these blood types. See the phenotype, blood type A. Okay, you can be IA, so you got 1 IA from mom in her egg and 1 IA from dad in his sperm. IA. Or you can be IA, little I. One parent had the IA and the other one had the little I for blood type O. Well, IA is dominant over little I, so you'll have blood type A. The same thing occurs for B. You either got IB from mom and IB from dad and you have blood type B, or you got an IB from one parent and a little I from the other parent, you're still blood type B because of the dominant recessiveness. IB is dominant over little I also. So IA and IB are both dominant over little I, and they'll be expressed. But when you have IA from one parent and IB from the other, they're both seen as blood type AB. That's codominance. They're both seen. So you have IA and IB antigens on the surface of your red blood cells. You're AB. Now this is what I am. I'm blood type O. Little I, little I. I have no antigen A nor B. So I'm blood type O. So you see the multiple alleles. This is definitely recessive. This is showing dominance and recessive. This is showing codominance here. And multiple alleles, IA, IB, and little I, multiple alleles. Let's do a cross. Here is an individual, blood type A, and they happen to be IA, little I. They're crossed with another individual, IB, little I. They're blood type B. Well, this individual can produce 2 different types of gametes, can't they? Half will have IA and half will have little I. And this parent can produce 2 gametes also. Half will have IB and half will have little I. So again, you use the Punnett square and put one parent's gametes across the top and the other parent's gametes down the side. And you add the boxes. IA, IB, IA, little I. And you put the big one first. IB, little I, little I, little I. Okay, so your genotypic ratio is going to be 1 IA IB to 1 IA little I to 1 IB little I to 1 little I little I. The phenotypic ratio, 1 type AB, that's the genotype, that's the phenotype, to 1 type A, genotype, phenotype, to 1 type B, genotype, phenotype, to 1 type O, genotype, phenotype. So again, they all work the same way. You know, you got the cross, set up the cross, get the gametes, do the Punnett square, get the genotypic and the phenotypic ratios. That's it. Now some variations in inheritance here. Let's look at a couple of these definitions here. Pleiotropy, the ability of a single pair, a gene pair, to have multiple effects. So this would be an example of sickle cell anemia, because you could have for hemoglobin, you could have the normal hemoglobin for mom and the normal hemoglobin for dad, and you have normal hemoglobin. You could have the normal hemoglobin gene from mom and the sickle cell from dad. You've got sickle cell trait. Or you could have the sickle cell hemoglobin gene from mom and the sickle cell from dad, and you have severe sickle cell. So there's 3 outcomes: normal sickle cell trait and severe sickle cell, which typically results in death. 3 outcomes. Epistasis, a condition in which one gene pair has the ability to mask or prevent the expression of another gene pair. And this is coat color and pigment deposition in mice. Now, I'll make up a sheet that will show you this. This is like coat color, like big B is black fur, little b is brown fur. And the pigment, big C is pigment, little c is no pigment. And we'll see how the pigment gene can control whether the fur is black or brown. I'll do one of those for you. Polygenetic inheritance. Many genes. A characteristic that is determined by the quantitative effect of multiple pairs of genes. Well, there are several things, polygenetic inheritance. Let's look at the next slide. This has one on there. This has to do with skin color. You see there are 6 genes here. 6 genes for this color. You see that over here is the lightest skin color. Here's the darkest skin color. And this is the average population here. We have the most. It's a combination. So over here you had all recessives for the lightest color. Over here you had all dominance alleles for the darkest. And right here you had 50-50. It's an average skin tone. This can be done for height also. This is the one I did in college, was for height. And they got a class of West Point cadets and lined them up. And they had the smallest, the shortest over here, the tallest over here, and the most numbers were in the middle. They were the same height. Like it could have been 5'6" to 6'3", and 5'10", which where most of the cadets stood. So it was a bell curve. When they were in lines, this was the longest line, and these 2 were the shortest lines, and the other ones were intermediates. So again, height is the same way. Hair color, eye color, skin color, height. There are a lot of them that are polygenetic, many genes. Other considerations, environmental impact on the phenotype. Can that happen? Yes. How about your dietary needs? Are they being met? If they're not, you're not going to experience your full potential as far as your body develops. There was something on the news and it showed this truck driver. And this truck driver that drives an 18-wheeler truck, he'd been driving it for 30 years, and it was showing him face on, and the left side of his face and his left arm was dry and wrinkled skin, and the right side of his face and his right arm were more smooth and less wrinkles. the sun coming in the window on the driver's side had an effect on his skin. It had dried it and made it more wrinkled from being exposed to the sun so much, more than the right-hand side. So that was definitely an environmental impact on his phenotype. Pedigrees, these are family histories, and a lot of times these are done on plants, but you can do it on humans too, to determine when a particular gene came into the population. So it says a family history for a particular trait, like where did that nose come in, or where did that ear shape come in, or where did we get dimples, or where did this disease come into our population, our gene pool. You can find that out the more information you know. Shows the relationship of parents to children over many generations. Let's look at some pedigrees over here and see if we can figure them out. And one of them we really can't yet because we haven't covered that yet. Well, they're all exposed here. This was a PowerPoint, but they're all exposed. This top one, you see that here's a couple that got married here. The circles are females, the squares are males. She is clear, he's darkened. Darkened means he's carrying something. Now, you look at their children, they had 6 children, so they had a lot of time on their hands, no TV, obviously. And when you look at the children, this female was affected, that male was affected, well, that male was not affected, that female was not affected, that female was, and that male wasn't. So it's asking, what was this man? Was he homozygous dominant, or was he heterozygous dominant? Well, it's kind of obvious he was heterozygous because half his kids got the gene that he's got in here for making it dark, and the other half didn't. So the trait, he was heterozygous for that particular trait, whatever it was. Down here, this is talking about marrying too close in the gene pool. You don't marry your first cousins. You don't marry them like so. You see that this couple, nothing wrong, have 3 kids, yeah. Their kids had kids, yeah. And 2 of their kids got married. There's not enough mixing of the gene pool. And look what happens. Some things happened. You carry lethal genes, and if you're marrying somebody that's different out of your gene pool, theirs are probably mixed up different than yours, and so these genes won't align. But there hasn't been a whole lot of gene shuffling in this gene pool, and they can line up and problems can occur. So when you marry too close to the gene pool, you can have some birth problems, some mental or physical problems. Now the one at the bottom, we haven't done this yet, it's X-linked or sex-linked. It's on the X chromosome. And here's a female, and she has a dot. That means she's a carrier. She has 2 X's. As long as she has 1 normal X, she's normal. And that's what this lady is here. She has 1 normal X, 1 affected X. Dad's got 1 normal, and then he got a Y. So they had a son. Well, Dad gave the Y, but Mom gave her affected X to this son, and he's affected. It's sex-linked, like pattern baldness, color blindness, those types of things. They had a girl. Mom gave her affected X to the girl. Dad gave his normal X. So she's a carrier. She's not affected. Mom gave her normal X. Dad gave his normal X. Mom gave her normal X. Dad gave his Y. Okay, so you can kind of see how that's happening. The mother carries a gene on her X chromosome that's not found on the father's Y. Examples: color blindness and pattern baldness. Now we have some human disorders here. Cystic fibrosis. Ever heard of that? A lethal genetic disorder. Homozygous recessive characteristic. So this is the homozygous recessive characteristic. You can have cystic fibrosis. So it's the recessive, homozygous recessive, lethal genetic disorder. Not good. Gonna die. This one called Tay-Sachs disease, a lethal disorder again. A recessive for a gene that regulates the breakdown of a class of lipids in the brain. Well, the individual's missing the gene for a particular enzyme, and so lipids build up in the neurons to toxic levels, and that individual's neurons eventually start dying. So, again, it destroys the nervous system. Lethal. Sickle cell disease, recessive gene for hemoglobin. This is only due to one amino acid out of thousands and thousands that does this. Recessive gene for hemoglobin. Heterozygous individuals are usually normal. They got the sickle cell trait. But homozygous sickle cell individuals show severe symptoms. That's severe sickle cell. So it does show pleiotropy. You can have normal. You can have normal affected, and that's going to be sickle cell trait. You can have affected, and that's going to show severe sickle cell. So it does show 3 outcomes. Another one, achondroplasia. Achondroplasia says heterozygous individuals show a type of dwarfism. Heterozygous is dwarf. Homozygous dominant individuals die. Homozygous recessive or normal. So the most common gene in the population is going to be, just 1 sec, is going to be homozygous recessive. Okay, it's going to be homozygous recessive. So the dominant gene is still in the population. It's just not the predominant allele in the population. If you watched Game of Thrones, you saw that Tyrion was a dwarf on there, and so he does have the problem there of having the heterozygous condition. Okay, Huntington's disorder, a lethal degenerative disease of the nervous system. The gene usually becomes active around 35 to 45. Now, this one is a death wish. Once you realize you have it, hopefully you haven't had any children, because you could have passed it to your children and they're going to die young also. From what I understand, there's some research. It's not been really published a whole lot, and they found the gene for this disorder. And there is a way they can do genetic therapy that will block this protein from being released. So they block the gene and the person can live a natural life. But this is under the experimental stages right now, to where they are blocking the defective gene to where that individual can actually have a normal lifespan. They still carry Huntington's. They could pass it to offspring, but they're not going to die early in life. Carrier recognition. This gets to doing amniocentesis. When people are having kids, sometimes the obstetrician or the doctor will say, do you want to have a karyotype? Do you want to have amniocentesis to find out if you have any genetics problems? And that's usually if the parent, the mother, is older. Now, it used to be 40, now it's 35 that they're asking for amniocentesis or some type of carrier recognition for the pregnant mother. Now, that's done to determine if they have a normal child or they have a child that potentially might have a birth defect like Down syndrome. And it's called fetal testing. And over here on the next slide, we'll look at these 2 types of testing methods. The one that was most common in the past is called amniocentesis. And this is a little bit dangerous because they have to use a needle to suck out some of the amniotic fluid to collect cells from that fluid that came from the baby. They will spin it down. They can look at the cells, make a tissue culture to get more cells, and several weeks later they can do a karyotype to determine if there's a birth defect like Downs or cleft palate or something like that, spinal bifida or whatever. Or they can go in and take some of the cells that are around the fetus and the sac, the chorionic villi, and they can do this karyotype the next day, karyotyping, to determine the same thing. So the amniocentesis and chorionic villi sampling is done just in case the female is getting up there in age and they want to make sure that their baby is okay. Because some people, if it's not, they will abort. Others, they won't do that.